Microarray is a tool used to measure the presence and abundance of gene expression in tissue. It is a pattern of ssDNA probes, which are immobilized on a surface called a chip or a slide. The chip or slide is usually made of glass or nylon and is manufactured using technologies developed for silicon computer chips. DNA microarray works by first collecting samples from a variety of organisms. For example of two samples, cancerous human skin tissue as a target sample and healthy human skin tissue as a control sample. Next is the isolation of mRNA. Extract the RNA from the samples using either a column or a solvent such as phenylchloroform. After isolating the RNA, we need to isolate the mRNA from rRNA and tRNA. mRNA has a poly-A tail, so we can use a column containing beads with poly T tails to bind the mRNA. And then rinse with buffer to release the mRNA from the beads. The buffer disrupts the pH and disrupts the hybrid bonds. The next step is create label cDNA. Add a labeling mix to the RNA. The labeling mix contains poly T, primers, reverse transcriptase to make cDNA, and fluorescently dyed nucleotides. We will add cyanine 3 fluorescent green, to the healthy cells and cyanine 5, fluorescent red, to the cancerous cells. The primer and RT bind to the mRNA, then add the fluorescently dyed nucleotides, creating a complementary strands of DNA. The next step is hybridization. This is where we want to detect specific DNA or RNA in sample. Apply the cDNA we have just created to a microarray plate. When comparing two samples, apply both samples to the same plate. The ssDNA will bind to the cDNA and already present on the plate. Lastly, detect the relative intensities of fluorescence under microarray scanner. The scanner has a laser, a computer, and a camera. The laser causes the hybrid bonds to fluorescent. The camera records the images produced when the laser scans the plate. The computer allows us to immediately view our results, and it also stores our data. All the things are involved in scanning and image processing. The use of microarrays for expression profiling is based on two fundamental principles. First, for many genes, a predominant factor underlying changes in expression is an alternation in the abundance of the cognate mRNA. And secondly, only DNA strands possessing complementary sequences can hybridize to each other to form a stable, double-stranded molecule. This type of analysis can be used to identify diagnostic or prognostic biomarkers, classify diseases such as tumors, monitor the response to therapy, and understand the mechanisms involved in the genesis of disease processes. Microarray also applied in the cancer research. Microarray-based technology is an ideal way in which to study the effects and interactions of multiple genes in cancer. As thousands of gene fragments can be located on an array, it can provide a genome-wide view of gene expression in cancer. It helps especially in the identification of single nucleotide polymorphisms and mutations, classification of tumors, identification of target genes of tumor suppressor, identification of cancer biomarkers, identification of genes associated with chemoresistance, and drug recovery. Gene microarrays have been used for cooperative genomic hybridization. In this technique, genome DNA fluorescently labeled and used to determine the presence of gene loss or amplification. Microarray-based expression profiling also allows us to identify families of genes as well as the important molecular and cellular events that might be important in complex processes like metastasis. Drug sensitivity. Another use for microarrays in cancer research is the investigation of the mechanism of action of drugs. It offers a new approach to studying the cellular pathways implicated in these mechanisms and in predicting drug sensitivity and unexpected side effects. This type of analysis will be beneficial for pharmaceutical companies in the development of improved drugs. Microarray technique has been known for its ability in detecting tumor formation. Thus, one of the examples of microarrays analysis in detecting cancer is in detecting breast cancer. This technique works by measuring the mRNA levels of the cell. Revelation of the remarkable degree to which variation in gene copy number contributes to variation in gene expression in tumor cells. Other than that, according to the study, microarray does use in detecting bladder carcinoma, melanoma, lymphoma, gastric carcinoma, and fallopian tube carcinoma.